All right. Welcome, everybody. Thank you for showing up to this presentation on the neuroscience of psychedelics. I'm so glad we have a full room today. It's a really exciting topic. Yeah, thanks for being here. A little bit about myself. My name is Ramzy. I studied neuroscience at the University of Michigan, and one of my most formative memories while studying neuroscience at the University of Michigan, I was an undergrad. It was my senior year of college, and I signed up for a seminar at the medical school for this group called the Center for Consciousness Science. And the Center for Consciousness Science, it's a interdisciplinary institution of different people from all over the university faculty. You have anesthesiologists. Well, the head is an anesthesiologist, because anesthesiologists, they're studying, people going in and out of consciousness. At what point do people go unconscious versus being conscious? And other people in the Center for Consciousness Science are physicists, mathematicians, grad students. And it was just a seminar where it was kind of like a journal club where people would nominate a paper about some mechanism of consciousness. Somebody would bring in a paper where they analyzed brain waves of people who are being put under anesthesia, and they kind of like try and pinpoint the point of, all right, when does this person go into unconsciousness? And stuff like that. So I was an undergrad, and this was a seminar held in the medical school, and it was mostly catered for grad students and medical students and PhDs, faculties, postdocs. But I was an undergrad, so I emailed the professor. I'm like, hey, I'm really interested in this seminar on the neurobiology of consciousness. Can I get an override? And he's like, all right, fine, I'll give you an override. So I got into the seminar, and one of the optional things you could do in this seminar was you could present a paper, and you could propose a paper, and you could deliver a presentation about it. And I don't know, I went out on a limb, and I found a paper that was just recently published by Dr. Robin Carhart Harris at Imperial College London. I would say about half of my presentation is his research. And the paper was called The Entropic Brain, which was a really deep dive on the neurobiology of psychedelics. And I kind of just went out on a limb and did this presentation on the neurobiology of psychedelics to this panel of some very esteemed anesthesiologists, physicists, physicians, postdocs, grad students. And I just went up on stage and started talking about psychedelics to all these people. And oh man, it was a really nerve-wracking presentation for me. I was sweating the whole time. But to my delight, afterwards, they ate it all up. We had a very vibrant conversation about, the brain waves and the brain networks and all the things that happen when people are put into brain neuroimaging devices on the influence of psychedelics. And it was kind of a really huge turning point for me, because it signaled that psychedelics were no longer kind of like relegated to the counterculture. Psychedelics had kind of entered back into the legitimate space of some very esteemed researchers in the medical and neuroscience field. And after that point, it really infused me with a lot of confidence and energy about, the legitimacy of this research. And we're currently going through a renaissance in the field of psychedelics, especially pertaining to neuroscience. And the psychedelic renaissance spans so many domains of culture. We have like the neuroscience community, the art community, the spiritual community, the indigenous community, you name it, the therapist community. So it's a really exciting time to be alive. I would say this is the 60s part 2. And yeah, so without further ado, let's begin the presentation. Okay, so before we can talk about the neuroscience of psychedelics, I just have to give a really short primer on neuroscience and neurotransmitters in general. So in the brain, you have nerve cells. They're called neurons. And the way that neurons communicate with each other is they don't really touch each other. So they're electrical signalers. They use electrochemical gradients to communicate, but unlike wires, neurons don't actually touch each other. There are some exceptions here or there that are kind of negligible that you don't have to worry about, but for the most part, neurons don't touch each other. So the way neurons communicate, the way one nerve cell transmits signals to the next nerve cell is it releases chemical signalers called neurotransmitters. And you've probably heard of some famous neurotransmitters. Serotonin is one of them. Dopamine is another one. And here, let me give you a nice little schematic. So right here you have, at the top you have a neuron that is an axon, and that long wire is called an axon, and it is connecting to another neuron. And if you zoom into one of these connection points, that is a close-up of that connection point, you see there's a gap. There's a gap between neuron A, neuron B. So neuron A is called the presynaptic terminal, neuron B is called the postsynaptic terminal. So the gap is called a synapse. So neuron A releases neurotransmitters, poof, they come out, and then they land on receptors on the second neuron. And this is the basis of basically how all of the signaling in your brain happens through neurotransmission like this. So one really useful way of looking at how neurotransmitters work is to consider a lock and key metaphor. So receptors on the postsynaptic terminal are only sensitive to molecules that have the right shape to activate them. For example, dopamine receptors are only sensitive to molecules that have the same shape as dopamine. Serotonin receptors are only sensitive to molecules that have the same shape as serotonin. So the metaphor that is most useful is a lock and key. So a lock is only unlocked by the key that has the right shape to activate it. And this is basically a really fundamental notion of how your brain kind of differentiates between different signals. So some more basics about neurotransmitters. So you've probably heard a lot about dopamine. You've probably heard a lot about serotonin. Those are really famous neurotransmitters. We'll talk a lot about serotonin. But believe it or not, 90% of neurotransmission in your brain is actually the neurotransmitters GABA and glutamate. So GABA and glutamate are the on and off switches in your brain. So glutamate is a excitatory neurotransmitter that is the primary, like on signal. So whenever your neurons have excitation, whenever they fire, it's because of glutamate signaling. Now the opposite, the converse, your neurons have off signals, and the off signal is GABA. And you'll find, a lot of, drugs and other substances that kind of like mellow you out, calm you down. They tend to be GABA agonists, meaning they have a shape that resembles GABA, so it kind of like triggers the same signal. So the other neurotransmitters that are more famous, like dopamine, serotonin, and the others, they represent actually a really small proportion of the neurotransmission in your brain, but the primary signalers are GABA and glutamate. Okay, so here's a video. This one's really cool. So this is how neurotransmission works. So there's a molecule, that's a neurotransmitter. It's landing on a receptor, and then boom, lands on the receptor and it activates it. So when it lands on the receptor, it causes ions to flow. And those ions, this will be really technical, the ions change the electrical gradient of the neuron. They either cause it to depolarize or to get hyperpolarized. So your neurons are constantly at a hyperpolarized state, meaning it's constantly pumping out ions to keep it at a gradient that is that is kind of like at a negative polarized state. And then when the right neurotransmitters land on receptors, it allows ions to flow. It changes the electrical gradient. Once it crosses a threshold, your neuron fires, and that's called an action potential. Okay, so the crucial system we're going to focus on today is the serotonin system. So you've probably heard a lot about serotonin. The unfortunate thing about the serotonin system is that it is really, really, really complicated. The serotonin system does so many things. You could fill entire encyclopedias with all of the functions of serotonin. One notable action of serotonin is it regulates mood, kind of like long-term affective states. It's also involved in appetite. It's involved in arousal. It's involved in cognition. There's a lot of serotonin in your gut. You know, you might find some statistics that say that something like 80 or 90% of the serotonin in your body is found in your gut. And that's true. However, one thing that you should note is if you ever see, like on Pop Science Twitter accounts or Instagram, if they talk about serotonin in your gut and stuff like that, just be aware that the serotonin that lives in your gut stays in your gut. Your brain makes its own serotonin. It has its own biosynthesis process to make serotonin in the cells in your brain. So the serotonin in your gut, even though like 90% of the serotonin in your body is in your gut, it doesn't cross the blood-brain barrier, so that stays in your gut. So if people say, oh, like your diet affects serotonin, yeah, it might in indirect pathways, but it really has nothing to do with the serotonin in your gut. But yeah, so the primary mechanism of psychedelics is they interface with the serotonin system, and the way they do that is they function as serotonin agonists. What that means is the shape of psychedelics, the shape of the molecules of psychedelics resembles the shape of serotonin. So therefore, it kind of like tricks the receptors into thinking that the psychedelic molecule is a serotonin. So I used the lock and key metaphor. The shape of the molecules of psychedelics resembles the shape of serotonin enough that it can trick the receptor into thinking that it's the right shape to activate it. So it lands on the receptor and activates it. And because it has subtle differences in shape, it can, change the downstream signaling. But in general, psychedelics work as serotonin agonists. So here's a little neat little tidbit in history. Psychedelics catalyzed a revolution in neuroscience. So I had just mentioned that there's a lot of serotonin in the gut, and scientists knew that. They had, dissected animals and stuff like that, and they had done, analyses, and they had found that, okay, serotonin is a molecule that is highly prevalent in the body, but they didn't know that serotonin was actually involved in cognition. They knew it was found in the brain, but they didn't realize it had a really prevalent role. But after LSD kind of was discovered and promulgated in the 50s and 60s, the chemists noticed a similarity in the structure of LSD and serotonin, and they kind of put two and two together. They're like, wait a second. So when people take this LSD molecule and it causes all these radical changes in consciousness, and then it highly resembles this molecule of serotonin, what if serotonin has something to do with the brain and cognition? And they were kind of able to have that aha moment. And that was catalyzed by the discovery of LSD. And prior to this, they didn't really have much insight that certain things like mental illness or neurosis or any of that stuff had anything to do with brain chemistry. They kind of just assumed it was all behavioral, or if somebody was like a bad kid, it was because their parents were shitty. So they never quite understood that chemistry, that molecules had a really profound influence on brain and behavior and affect, and it was actually LSD that kind of ushered in that realization. So when we talk about psychedelic drugs, there's a bunch of them, but they tend to fall within two primary categories. So the two primary categories of molecule shape that psychedelics tend to fall into are the tryptamines and the phenethylamines. And they're both monoamines. So what that means is, do you see that tail? That is an amine, and there's one of them, so they have one tail that makes them monoamines. So in neuroscience, monoamines are a very prevalent category. So Serotonin, dopamine, norepinephrine, all those neurotransmitters, they're also monoamines. But these are the two categories that most of the drugs are going to fall in. I really like this slide a lot because it really just shows how similar the shapes of the molecules are. So at the very left, you have the base structure. So tryptamine is the base structure. It is one step derived from tryptophan, which is an amino acid you find in your diet. And you do subtle modifications to the tryptamine base structure, and then you get all of these different molecules. So if you look right here at the top, you have serotonin. So the chemical name for serotonin is 5-hydroxytryptamine, so 5-HT. So this phrase right here, 5-HT, remember that. Remember 5-HT, because that's going to come up a lot. And 5-HT is simply the chemical name for serotonin. So the reason, I'll break it down for you. 5-hydroxytryptamine. So if you look at the tryptamine molecule, each one of the little spots on the ring has a number. So at the 5 point on the ring, you have a hydrogen and an oxygen. That makes it 5-hydroxytryptamine. It's really simple as that. Down here, you have psilocin and psilocybin. So it's basically the same base structure as serotonin, except here at the 4 point, you have a phosphate group if it's psilocybin. DMT, dimethyltryptamine. So at the end of the tail, you have two methyl groups, dimethyltryptamine. 5-MeO-DMT, this is in the toad, bufo. At the 5, you have a 5-methoxy group, so 5-MeO-DMT. It really is as simple as that when you break down the structure of the molecules. And then here at the very right, we have LSD, which is a really complicated molecule, which explains a whole lot about the differences in how how LSD affects people. It lasts a lot longer, and it affects a lot more neurotransmitter systems than just serotonin. I think LSD has the base structure of tryptamine and phenethylamine in it, so it's a really unique molecule. But the premise, the main point here is that all of the psychedelics have the same exact base structure as serotonin, which ultimately derives from the base structure of tryptamine. Here's a really cool slide that I also really like. It shows all of the molecules in their 3D form. And yeah, you can kind of see how a lot of them have really close resemblance in their shape. And right here on the side, LSD is a lot more complicated. But I always like to see the visualization of the molecules just to see how cute they look, and they're pretty cool. One of my dreams is to have like little 3D models of the molecules just like on my desk as paperweights. I think that'd be a pretty cool gift. Hint, hint. So there are so many tryptamines. The list is huge. So you have bufotenin, DMT, melatonin, the thing that makes you sleepy, is also a tryptamine produced in the pineal gland. 5-MeO-DMT, psilocybin, psilocin, serotonin. The list goes on and on and on. And basically what you do is you go back to that base tryptamine structure, and at different little spots along the rings, you slap on a methyl group here, you have a tryptamine molecule. You slap on a hydrogen and oxygen here, you have another tryptamine molecule. So if you just like do subtle modifications to that base structure, you get entirely new molecules. And that's what these are. Okay, so the second category is the phenethylamines. So the phenethylamines have an entirely different structure. So the tryptamines, if you recall, their structure really closely resembles that of serotonin. The phenethylamines, their structure actually more closely resembles that of dopamine. But through some weird, biochemistry, the phenethylamines can still activate serotonin receptors, even though their shape more resembles that of dopamine. But, you'll notice that a lot of the drugs in this class have a similar shape. So you have the first one we have here is dopamine. DA stands for dopamine. Mescaline, that's the classical psychedelic that's on here. You'll see its shape really closely resembles that of the other phenethylamines. Here we have methamphetamine and amphetamine. So amphetamine, this is more colloquially known as Adderall. You might have heard this before, but amphetamine is basically diet methamphetamine. So methamphetamine has an extra methyl group, and what this does is, so the methyl group makes it more lipophilic, which means it's more lipid soluble, and neurons have a lipid bilayer, and things that are more lipid soluble are more able to cross the blood-brain barrier. So the main difference between methamphetamine and amphetamine is that amphetamine is just simply more lipid soluble, crosses the blood-brain barrier more rapidly, therefore it's more potent. So yeah, Adderall is basically diet meth, and MDMA and MDA are basically methamphetamine and amphetamine with a methylene dioxy ring attached to it. So MDMA stands for methylene dioxy methamphetamine. MDA, sassafras, stands for methylene dioxyamphetamine. So they're basically a methamphetamine molecule with this little extra piece attached to it, and MDA is an amphetamine molecule with that extra piece attached to it. I find it so fascinating that all these drugs are just subtle variations of the same base shape. And here's another little slide with the 3D molecules rotating. So there are so many different compounds you can derive from the phenethylamine structure. Hundreds of them. If any of you have heard of the N-Bohm series, those are all phenethylamines. If any of you have heard of the 2C series, those are all phenethylamines. Sasha Shulgin was a huge fan of the 2C series. He synthesized a lot of the 2C molecules like 2CB, 2CE. They're all phenethylamines. They're all subtle modifications of that same base structure. And if you are a chemist, a really talented chemist like Sasha Shulgin, you can sit in the lab and just go crazy with it and just come out with hundreds upon hundreds of novel molecules that are just modifications of that same base structure. And that's how he was able to do it. I mean, he was a wizard, but he wasn't really a wizard, because all you do is just you have that, once you have that base structure, you just tweak it. You just go back and rinse and repeat, and you do one tweak and you test it out, and if you don't like it, you go back to the lab and you tweak it again, and it's really simple stuff, honestly. Okay, so back to the basic mechanisms of how psychedelics exert their influence on the brain. So I mentioned this earlier. So the shape of the psychedelics, because their molecule has a chemical structure that so closely resembles that of serotonin, it can land on the serotonin receptor and fall into the right grooves to kind of imitate serotonin, and it activates the receptor. So right there, you have an image of serotonin in its receptor. And then down here, you have an image of LSD in serotonin's receptor. So it's like the lock and key. So the molecule, its shape is just so on point that it can land in the receptor and fool it. And then here is another 3D. These are kind of very hastily rendered 3D models of the receptor. This is a more faithful representation of what the receptor looks like. It's a bunch of different proteins bound together, and then there is the LSD molecule inside of the receptor. Yeah. Okay, that's a great question. So the question is, different psych meds versus psychedelics, how do they affect the serotonin system? So psychedelics are postsynaptic agonists, meaning they land on the receptor, the serotonin receptor, and trick it into activating. A lot of psych meds, like for depression, such as SSRIs, selective serotonin reuptake inhibitors, they actually work at the presynaptic level. So if we go all the way back to this diagram. So the way that like SSRIs work is you have the presynaptic cell that releases serotonin, and then after the serotonin is done being used, there's a process that happens called reuptake. So that basically, what that is the serotonin that was just being used by the synapse, by the presynaptic cell, it gets recycled. So after it's done, doing its neurotransmission or whatever, it will fall off the receptor back into the synapse, and then the first cell A, the presynaptic terminal, will suck it back up like a vacuum cleaner in a process called reuptake. And then it'll bring it back in and recycle it. So the way that SSRIs work is they block reuptake. So it blocks the first cell from absorbing the serotonin back into it. And what that does is it makes it so that there's the serotonin sticks around in the synapse longer and it has another opportunity to land on a receptor and activate it and do that signaling. So psychedelics and certain meds like SSRIs, they have very different mechanisms. Let's go back. Okay, so I mentioned this earlier. So LSD is a fascinating molecule because it activates so many more receptors than most of the classic psychedelics. So most of the classic psychedelics activate so remember 5-HT stands for serotonin, 5-hydroxytryptamine. So I'll just say this. So a lot of times people will say something silly. They'll say the serotonin 5-HT2A receptor, but 5-HT just means serotonin. So they're basically saying the serotonin 2A receptor. Don't do that. So I'll just say the serotonin 2A receptor, the serotonin 2C receptor, and the serotonin 1A receptor. So most of the tryptamines activate just those three receptors. The phenethylamines will activate the 2A and the 2C. LSD activates all of these receptors. So you have a bunch of serotonin receptors that it activates. Just to clarify that there are lots of different variations in serotonin receptors that do different things. The receptors have subtle differences in their molecular structure. And it's too complicated to get into all the details, but the main thing to take away is that there are different subclasses of serotonin receptor. So LSD activates all these different serotonin receptors, and then it also activates dopamine receptors as well. So you have D1, D2, and D4, which is a really interesting thing because if we want to talk phenomenology, a lot of people describe LSD as an experience that tends to be more stimulating. People have more cognitive control, and that might be entailed by the fact that LSD does have influences on some dopamine receptors. But even though I just talked about how all the psychedelics activate so many of these different receptors, and LSD has a much larger repertoire, the main psychedelic effects are exerted by one type of serotonin receptor. That's the 2A receptor. So the 2C and the 1A receptor, the activation of those receptors is going to have it's going to color the experience in some subtle ways, but the main warrior, the main reason why psychedelic experiences are felt in the brain is through the activation of the 2A receptor, and that's crucial. And the way they know that is they'll have a special drug that blocks the 2A receptor in specific, and they'll give people that drug. So this drug will only block the 2A receptor. It won't block the 2C or the 1A or any of the other serotonin receptors. It'll only block the 2A receptor. And then they find that if you give people a drug that blocks the 2A receptor, and then you give them a psychedelic like LSD or psilocybin, they will not trip. Okay, cool. So this is the star of the show, the serotonin 2A receptor. This is why we trip. So that is a illustration of what the 2A receptor looks like with a LSD molecule bound to it. So the 2A receptor, it is found in regions of your neocortex that are involved in high-level processing. So these are the regions of your cortex that are taking in lots of signals from throughout your whole brain and kind of like aggregating and doing a lot of the calculation and a lot of the really post-secondary processing. So this is really down the line. So a lot of the highly refined signals in your brain are going to be entailed through signaling with the 2A receptor. And there is a special layer of neurons. So your neocortex has 4 layers of neurons. And the deepest layer, which is a layer 5, that is where your 2A receptors are most densely located. So layer 5 of your neocortex is where a lot of the really high-powered, high-level calculatory action happens in your brain. And there's a special type of neuron called pyramidal neurons that these receptors are most prominently expressed on. And what happens when the 2A receptor is stimulated is it causes the cell to depolarize. Earlier I had mentioned that your neurons are kind of like always held at a polarized state. And it takes a lot of energy for your brain to kind of like hold your neurons at that polarized state. And then when your receptors are activated, it opens ion channels, and those ions flow into your neurons, and it changes the electrical gradient. And once that electrical gradient crosses a threshold, your neuron will fire. Here's what it looks like for your neuron to fire. Somebody stuck a little camera in someone's brain. Okay. So I spent a lot of time talking about LSD, but let's pivot to psilocybin real quick. So we all know psilocybin mushrooms, but the thing is, so psilocybin is a molecule that's found in the mushrooms, but psilocybin is actually not what gets to your brain and what activates your receptors. So when you consume psilocybin, there is a phosphate group right there. If you look at the molecule, you can have a laser pointer. If you look at the molecule, so this is the tryptamine base right here. That's the amine, so the monoamine. So right here, you have a phosphate group. And then once you consume psilocybin through some biochemical process in your body, the phosphate group gets dephosphorylated and it becomes psilocin. So psilocin is actually what enters your brain and stimulates your 2A receptors. And here's a neat little thing I'll show you about psilocin. So these are actually methyl groups right here. So if you were to remove this hydroxy right here on the psilocin molecule, that's DMT. That's dimethyltryptamine. So psilocin is 4-hydroxy dimethyltryptamine. So that's a really neat thing. A lot of people say that if you take high enough doses of mushrooms, you enter a space that's really similar to the DMT space. And what's really interesting is that the psilocin molecule literally is a DMT molecule with a hydroxy on the little 4 segment of the ring. And here's another neat little thing. There are other prodrugs that also get converted into psilocin in the body. So one of them is 4-ACO-DMT. So I say 4-ACO-DMT. So when you consume 4-ACO-DMT, this acetyl group gets spliced off, and it also becomes psilocin. Here's another really interesting thing about 4-ACO-DMT. In many places in the United States, it's not scheduled. You could actually buy this online. There are research chemical websites where they sell 4-ACO-DMT, and it's completely legal. Last I checked, it's not a scheduled substance. So in many ways, if you, for some reason, you can't find mushrooms or you don't want to grow your own and you want to have a psilocin experience, you could acquire 4-ACO-DMT legally, consume it, and then have a mushroom experience, a psilocin experience. And people describe the experience from the difference between taking like mushrooms versus taking 4-ACO-DMT is people who have done both, they'll describe them as identical. They'll say that 4-ACO-DMT is like taking mushrooms but with no nausea. So pro tip. So this is some really fascinating research that has been published recently in the past few years, coming out of the Jima Borjigan lab at the University of Michigan, which is my alma mater. I actually know the people that work in this lab. They're doing really amazing research. So they have found that DMT is present in the mammalian brain. So this is something that has always been hypothesized. So if you watch documentaries like DMT, the Spirit Molecule, stuff like that, or read the book, researchers like Rick Strassman have kind of like always put forth this hypothesis that DMT is found in the brain, you produce it, and he hypothesized it's in the pineal gland. And there's a whole lot of mythology regarding the pineal gland and its role. Descartes thought the pineal gland was the seat of the soul. Pineal gland is where melatonin is released, and melatonin is what signals your body to start getting sleepy. So there's this kind of like interface between wakefulness and sleep, consciousness and unconsciousness, and you can kind of like take that archetype and blow it up and say life and death and the soul. But so there's a lot of mythology about the pineal gland. So Rick Strassman kind of originally proposed this back in the 90s, that the brain produces its own DMT, and it gets released when you dream, when you're giving birth, when you die, when you have an extremely traumatic experience. The thing is, Rick Strassman proposed this as hypothesis. This wasn't quite fully established in the scientific literature. But what we have been seeing coming out of the Jima Borjigan lab at the University of Michigan is they're actually finally proving some of these hypotheses. One of them, namely being, is that DMT is actually found in the mammalian brain. So they used rats. They studied rats, and they were able to discover that, yes, DMT is found in the rat brain at levels similar to other neurotransmitters like serotonin. So it wasn't trace amounts. They didn't just like analyze a rat brain and find like little trace molecules of DMT. No, they actually found that the levels of DMT in the rat brain were comparable to levels of other neurotransmitters like serotonin. And so they didn't want to just find, is there DMT in the mammalian brain? They wanted to like determine, all right, if there's DMT in the mammalian brain, how does it get there? Because with all the other neurotransmitters, scientists have really detailed accounts of how they are biosynthesized in the body. For example, serotonin is synthesized in the body. You eat tryptophan in your diet. Tryptophan is an amino acid that is found in lots of food. So you eat tryptophan, that tryptophan becomes tryptamine, and that tryptamine becomes serotonin. And there's all these little steps along the way. So these researchers, they wanted to tease out, all right, if DMT is in the body, how is it synthesized in the body? So they were able to kind of pinpoint two molecules that are required for the biosynthesis of DMT, and they wanted to show that where DMT is present in the brain, are there also present these precursor molecules that are that can be part of the process of the biosynthesis of DMT, and they were able to do that. So they found the mRNA of INMT and AADC. The full scientific names of these substances eludes me at the moment, but those are two molecules that are required for the biosynthesis of DMT, and the thing is, they have to be found together. So DMT can't be synthesized in the body with just one of them. You need to have both of them present in the same place, and they were able to find regions of the brain that have both of these precursors required for the synthesis of DMT in the brain. And those regions included the cerebral cortex, the pineal gland, and the choroid plexus. But here's the crazy thing. So one thing they were able to do was show that DMT, so what they did was they induced cardiac arrest in these rats. So they basically killed them. And they had little devices that were analyzing the levels, the concentrations of certain substances, and they were actually able to show that at death, there was an increase in DMT levels in V1, the visual cortex. So in the 90s, Rick Strassman had hypothesized that at death, DMT is released in the brain. The researchers were able to show that. When they induced cardiac arrest into the rats, there was a secretion of DMT in the visual cortex, which was really amazing discovery. But here's the crazy part. They had rats who had intact pineal glands. They had other rats where they had removed the pineal glands. And the secretion of DMT at death was the same whether or not the rats had a pineal gland. So there's this kind of overarching mythos that DMT and the pineal gland have this, intricate relationship. The researchers were able to show that no, not really. The pineal gland is not necessary for the secretion of DMT at death. So even if the rats had their pineal glands taken out, they still released DMT at death. So that's another thing. So they were able to prove one of the theses, one of the hypotheses of Strassman, and they were able to debunk another. Okay, so who's heard of ayahuasca? So if you just have molecular DMT, that whitish, that yellowish powder that people smoke, you cannot put it in a capsule, swallow it, and trip. You just can't. And that's because there is an enzyme called monoamine oxidase. I had mentioned monoamines earlier on. So the monoamines were the molecules that have that one tail. So monoamine oxidase is an enzyme that metabolizes all of the monoamine molecules in the body. So this one molecule metabolizes dopamine, serotonin, norepinephrine, DMT, all of them. And what I mean by metabolize is after the neurotransmitter has done its job on the receptor, it'll enter back into the synapse, and then monoamine oxidase will land on it and kind of like metabolize it so it's no longer active. So when people try to consume DMT, the monoamine oxidase, which is found all throughout the body, will immediately latch on to the molecules and just deactivate it. So this is why, in order to experience DMT orally, and this is why smoking DMT lasts a very short time, because when you smoke DMT and then it lands on the receptors in your brain, the monoamine oxidase is already getting to it. It's already attacking the DMT and neutralizing it. So people say that DMT experiences last, 3, 4, 5, maybe up to 15 minutes. That's because as soon as they smoke it, the monoamine oxidase is neutralizing it. It's metabolizing it. So there are certain molecules, substances you can take called monoamine oxidase inhibitors, MAOIs, that they prevent the degradation of the monoamine molecules. And an example of a MAOI is harmaline. Another one is harmine. I'm trying to think of the scientific name that encompasses both of them. Beta-carbolines or something like that. Yeah. But yeah, so when you consume an MAOI, and then this enters your body and it neutralizes all of the monoamine oxidase, And it allows the DMT that you consume orally to actually become bioactive. And this is how ayahuasca works. So you have to couple the DMT with an MAOI in order for it to go through your system, into your bloodstream, and then into your brain without it being degraded. But here's the catch. So the MAOI does not only prevent the degradation of DMT, it also prevents the degradation of dopamine, serotonin, norepinephrine. So it's kind of like all of the monoamine neurotransmitters are prevented from being metabolized. So when people consume a MAOI, let's say ayahuasca. So when people have an ayahuasca experience, that's not simply a DMT experience. When people consume ayahuasca, they have, so DMT is going to be stimulating the 2A receptors, but also you have elevated levels of dopamine in your synapse, elevated levels of serotonin in your synapse, and elevated levels of norepinephrine in your synapse. So ayahuasca is an entourage effect of dopamine, serotonin, norepinephrine, and DMT. That's why ayahuasca experiences are very different than the experiences people have just by smoking DMT. Oh, and just a little tidbit of safety. Because MAOIs also inhibit the degradation of serotonin, if people are taking SSRIs or any drug that elevates the levels of certain neurotransmitters in the brain, it can be dangerous to couple them with SSRIs. Because that can cause serotonin syndrome. It just makes it so that there is just so much serotonin in your brain, and none of it is being metabolized at a rate that it should, and that can be pretty dangerous. It can be life-threatening. So just be mindful that SSRIs and MAOIs do not mix well together. So the question is there any danger in combining things like MDMA or DMT and tryptamines and phenethylamines? Is that right? When it comes to mixing drugs, any sort of mixture has its own inherent dangers. So it's really hard to answer that question really effectively. But if I can say anything about this, the dangers of mixing those drugs is entirely different than the dangers of mixing those drugs with MAOIs. So the MAOI mixture is the more dangerous one, I will say. And primarily MAOIs with MDMA don't. MDMA, we'll get to MDMA. MDMA, that's actually the next slide, so we'll talk about that right now. So MDMA is not technically a classical psychedelic. The classical psychedelics are LSD, psilocybin, mescaline, and DMT. And MDMA has an entirely different mechanism. But certain organizations like MAPS, Multidisciplinary Association of Psychedelic Studies, they've been getting a lot of PR for their research with MDMA and how it's useful in treating PTSD, especially in veterans. So even though MDMA is not a classical psychedelic, its therapeutic uses kind of overlap with that of psychedelics. So it deserves a mention. So I'll just talk about how MDMA works. It's actually a very different mechanism than the other psychedelics. So I had mentioned that the other psychedelics work as postsynaptic agonists, meaning, so here's the receptors. These are serotonin receptors. So the way classical psychedelics work is their molecules will land on the receptors and tricking them into thinking they're serotonin. The way MDMA works is completely different. So I'm glad somebody asked the question about SSRIs earlier because I got to broach the topic of reuptake. So this is called a serotonin transporter. So this transporter is on the presynaptic terminal, and I had mentioned that the transporter does reuptake. So after serotonin is done being used by these receptors, they'll plop off, and then the transporter will suck it back up like a vacuum cleaner so it can be recycled. And then once it's sucked back up, it'll enter back into the cell where it'll be stored in these little storage units called vesicles. And this molecule called VMAT2 is it's a vesicle transporter that basically takes the serotonin that's inside of the cytoplasm of the cell, and then it'll suck it back in here for storage. And then when you have natural organic neurotransmission, you have a thing called exocytosis, where the vesicles will kind of come out to the edge of the neuron, and they'll kind of like blend into the wall like a bubble, and then it'll release the neurotransmitters. This is how natural organic neurotransmission happens all the time in your day-to-day regular consciousness. But back to the transporter. So the transporter is a vacuum cleaner. So the way that SSRIs work is it blocks the transporter, so the serotonin can no longer be sucked back up into the cell. So the way that MDMA works is, number one, it lands on the transporter and then stops it from sucking back up the serotonin, but has another effect, which is bananas. It'll actually make the vacuum cleaner start pumping in reverse. And then another thing that it'll do, so I had mentioned that VMAT is this little gateway, this little molecule that takes the serotonin and puts it in storage. It'll land on the VMAT and cause the cell to start dumping its entire supply of serotonin into the cytoplasm. So imagine this. You have all the serotonin in storage inside of your cell. All that serotonin in storage gets pumped into the cell, and then the transporter that's usually sucking serotonin into the cell starts pumping in reverse, and it basically floods your synapse with a lot of serotonin. And that's how MDMA works. So one interesting thing to note is that psychedelics work by tricking the serotonin receptors into thinking that it's serotonin. And then because the shape is subtly different, it causes differences in the secondary signaling in the cell. But the way that MDMA works is it causes your cell, your presynaptic cell, to just flood the synapse with real serotonin. Yes? Thank you, great question. So a lot of people report that after having an MDMA experience, the next day you'll have a hangover. And I think that explanation very thoroughly explains why people have a hangover. Because you have all the serotonin that's just like chilling in your cell in storage waiting to be released. And then with MDMA, all that serotonin that is in your cell that is hanging out in storage just basically gets flooded out. It gets released. So your serotonin reserves in your cell just all get depleted. So the next day, when you feel the hangover, that's because all of your serotonin is gone. It's been depleted. And then, of course, your cell's going to make more. It's going to it has its own biosynthesis process where it's going to generate more serotonin. But that takes time. And that's why they say that 5-HTP is a good supplement to take to kind of help ease the hangover, because 5-HTP is one of the precursors to serotonin. And by taking that precursor to serotonin, you kind of like equip your body with more of that precursor so it can kind of like expedite the process of generating new serotonin. But yeah, that's why you have a hangover, is because all of your serotonin reserves have been depleted. And that's why, real quick, that's why taking MDMA with an MAOI, a monoamine oxidase inhibitor, is so dangerous, like really, really dangerous, because you're flooding your synapse with so much serotonin, and if you take it with MAOI, that serotonin cannot be metabolized. It cannot be degraded. It cannot be neutralized. So you have all this serotonin that is just flooding your brain, and it's staying there, not being degraded, and you can get serotonin syndrome, and you can die. Yes? No, that's only if you couple it with oh, I get your question. Eventually the MDMA wears off, and then your cells will be able to reuptake it. But the thing is, if you So no MAOI. If you take it regularly, the MAOI is still going to metabolize that serotonin, and then once it gets metabolized, it gets carried away, and then it can get recycled into the cell through other downstream mechanisms. So it's not like your serotonin is gone forever. It's just for the temporary window after the fact, your brain, your cells, your presynaptic cells are just depleted of the supply. Yes. Yeah, so all of these benchmarks that people say you should wait a month or a week or 6 months after MDMA, none of those have like really robust scientific reasoning for those. Those are just like numbers that somebody made up as, you know, just like a ballpark. But I will say that when it comes to MDMA versus other psychedelics, MDMA does not have the same safety profile as other psychedelics. I didn't include any slides in here about the case studies of people who have taken monstrous overdoses of LSD. I don't want to get too off track. Let me check. How are we doing on time? Okay, I think we have enough time. So there are case studies of people who have taken monstrous overdoses of LSD. For example, there was this one woman who was digging through her roommate's drug stash and found a baggie of white powder that she thought was cocaine. It turned out the baggie was pure crystalline LSD. And if you know anything about LSD, an active dose is in the micrograms. So 100 micrograms. So 100 micrograms is 1/10 of a milligram. A milligram is a speck of sawdust. So imagine a speck of sawdust and take 1/10 of that speck of sawdust. That is one full dose of LSD. A speck of sawdust is 10 doses of LSD. Imagine snorting a line of this white powder you found in a baggie. So it turned out to be 55 milligrams. 55 milligrams of LSD. That's 550 hits, 550 doses of LSD. Imagine taking 550 Tylenols. Your liver is gone. Imagine taking 550 shots. You're dead. You're dead. But this woman, she snorted 550 hits of LSD, and I'm not gonna lie, it was a grueling journey. I don't know the exact time scale, might have been 48 hours, maybe 72. I think it was 48 hours. Somebody can correct me on that. And she was like hopping in and out of consciousness. She was like frothing at the mouth, and but after it was all over, she was completely fine. Completely fine. Made a full recovery, 100%. Like, the way that psychedelics work physiologically, they don't affect any of your, crucial physiological systems. For example, opioid drugs, they depress your respiratory system. So if you overdose on an opioid drug, you will stop breathing and you will die. If you overdose on cocaine, it activates your sympathetic nervous system. Your heart will start beating like crazy, and your heart will give out. You will die. With psychedelics, they don't affect your brainstem. So there is basically no known dose. The LD50 is probably something really high, and they figure out the LD50, which means the lethal dose for half of the animals that consume that amount. But the LD50 for humans for psychedelics is just so high. It's basically impossible to overdose on a psychedelic like LSD or psilocybin or DMT. However, with MDMA, just to come back around, MDMA does not have the same safety profile as the other psychedelics. You can overdose on MDMA and you can die by taking too much MDMA. And forget dying. MDMA can also be neurotoxic if you take too much. So there is some dubious research, controversial research. For example, some of the studies about the neurotoxicity of MDMA, one of the researchers who published one of the papers, it turns out he had mistaken MDMA for methamphetamine. So he published this article that talks about how MDMA is neurotoxic and stuff like that. It turns out he actually had accidentally given his animals meth, not MDMA. So that research was debunked. But there is still some research that shows that taking as much as 2 times the standard dose of MDMA can be neurotoxic. And what that does is it causes death of the serotonin terminals in the brain. So I'm not trying to sound any alarms, or maybe alarms should be sound, but just be careful with MDMA. It does not have the same safety profile as the other psychedelics. In terms of minimizing the refractory period or whatever terminology I'll use for how long you should wait after consuming MDMA, there isn't really anything scientific to say about it. Just be responsible, take it in moderation, and 5-HTP is good to help replenish the reserves, but MDMA is a substance that more care should be considered, and there's like a whole bunch of supplements that can be taken to kind of like alleviate, the after effects that I saw hand. Very similar. MDA and MDMA are very similar. It's very similar, yes. Okay, so great question. So basically, a lot of the research coming out about MDMA and its use in therapy and treatments for PTSD, it shows that has a very high rate of basically wiping out PTSD. I'll give a little quick commentary about what might be happening psychologically. Every time you recall a memory, that memory gets reconsolidated based on what happens when you recall it. We never recall memories as they happened. When you recall a memory, you're just recalling what was reconsolidated the last time you recalled that memory. So if people recall memories that are very traumatic and fearful and scary, that emotional valence is going to stay encoded, because if you have a traumatic memory, a traumatic experience, and then you re-experience that memory, and then those same emotions of fear and horror and terror are there, when that memory gets reconsolidated, that same coding is going to stay. When people have, let's say, therapy with MDMA, and they are sitting with a therapist and they're talking it out, and they are recalling these traumatic experiences. When people are under the influence of MDMA, instead of having the memories just coded with fear, horror, terror, they can also experience things like compassion, hope, love, forgiveness, self-compassion, all these things. So when that memory gets reconsolidated, you have all these new emotional valences that are going to be coded with that memory. So if a person has a traumatic memory, let's say you're a veteran and you are reliving seeing your friend die, or you're reliving shooting somebody and killing them, and then you're haunted by that. And then having an experience with MDMA, when you're sitting through and recalling that memory in that space, you're now imbuing that memory with compassion, with forgiveness, with love. And then when that traumatic memory, every time you recall a memory, it gets reconsolidated. You're going to reconsolidate that memory with those new emotional valences of forgiveness and love and hope and compassion. So that might be a psychological mechanism that explains why it's so effective at kind of like pulling the plug from the vicious cycle of trauma. Any more questions? Yeah, that's a good question. I don't recommend taking 5-HTP before an MDMA experience because that might potentiate it and make it more powerful. There might be a ceiling. I don't have the requisite research to give you like a solid answer on that. But I don't recommend that. Cool, I think we should move on. Oh. Okay, so ketamine. This is also not a classic psychedelic. It's an oddball, but in recent years, ketamine has been gaining a lot of fame for its use in treating depression. It's a dissociative anesthetic, but it can also induce psychedelic-like states. So ketamine does not affect the serotonin system at all, and that's what makes it an oddball. So I'd mentioned earlier on that one of the primary signalers in the brain is glutamate. Glutamate is the primary excitatory signaler. So every time your neurons fire, every time you get that on signal, every time you get an action potential, that's usually promulgated by glutamate. So what ketamine does is there is a special type of glutamate receptor called the NMDA receptor, and ketamine will actually enter into the receptor and block the ion flow. So it'll basically block the NMDA receptor from firing. And by blocking the NMDA receptor, it blocks some glutamate transmission, and that is how you put people into anesthetic states. So when you give people enough ketamine, it stops their neurons from firing. And by stopping the neurons from firing, you knock people unconscious, and that's how you get anesthetic states. And when it comes to lower doses of ketamine and how that might exert antidepressant effects, this gets really complicated really quickly. But I'm not going to go all the way back to the first slide, but there's a diagram of a neuron landing on another neuron. So the presynaptic neuron, the neuron A, actually has other neurons projecting to it that are called GABAergic interneurons. And then ketamine might be blocking the transmission of the GABAergic interneurons, which basically inhibit the neuron A. So if you're inhibiting the inhibitor, you get disinhibition, and that's how ketamine might be exerting its antidepressant effects. There's some really complicated neurobiology with ketamine. I don't want to get too much into it, but yeah, it works on the glutamate system, and we got bogged down, so I'm going to try and be really quick. So LSD, why does it last so long? So most psychedelics do not last 12 hours like LSD does. And there's some new research that just came out that discovered why LSD lasts so long. So they actually found that there is a section of the LSD molecule that when it lands on the serotonin receptor, so I'll actually point it out to you. Do you see this little section of the LSD molecule right here? So that is a diethylamide. So there's two of them. So ethylamide, so two of them, diethylamide. So this section of the LSD molecule, lysergic acid diethylamide, actually functions as like a little grappling hook. It's like a claw. So when LSD lands on the molecule, the diethylamide kind of like hooks on to a little flap on the serotonin molecule, and it just doesn't let go. It just like hooks onto it. And typically when molecules land on receptors, eventually they'll do their job and they'll plop off and they'll get metabolized. But because LSD has this hook, it has this grappling hook that grabs on to a little flap on the lid of the receptor, it's unrelenting. And then the LSD will not be metabolized. So typically what has to happen is the neuron actually has to absorb the receptor into the cell to metabolize the LSD. And this usually takes 12 hours. So that's why LSD lasts a lot longer than the other molecules. It's because a physical property of the molecule itself has this little hook, this little claw that grabs onto a lid on the receptor and stops it from being unplugged. Okay, so early on, I had mentioned that one of the ways that scientists discovered that the 2A receptor is the primary mechanism of psychedelics is they would block, they would have drugs that block the 2A receptor, and they found that if you gave people a psychedelic after blocking the 2A receptor, they would not trip. So there are commonly prescribed pharmaceuticals that are actually, that do just that. So trazodone is one of them. Trazodone is very commonly prescribed as a sleep aid and an antidepressant. It's a very commonly prescribed pharmaceutical, and it works as a 2A receptor antagonist. So this whole time I've been talking about agonists. Agonists activate the receptors. Antagonists block the receptors. So trazodone, in many ways, is a trip killer. So of course people talk about other pharmaceuticals that can be used as trip killers. Some people will take Xanax or other things to kind of like end a trip. But with taking benzodiazepines and other drugs like that, killing a trip through that means doesn't just kill a trip, it also just like knocks you out and does what Xanax does. But the way trazodone works is it's a trip killer without like the rest of the depressive effects. So this is a really cool brain imaging diagram that basically shows the relative concentration of where the 2A receptors are found in your brain. So if you look at this little key right here, you have the cooler colors is where there's not very many 2A receptors, where there's not a high concentration of 2A receptors. And then, as it gets warmer, all the way up to the red, that is where the highest concentrations of 2A receptors are in the brain. And I think this is a really cool diagram because it kind of like shows you anatomically like where what regions of your brain are mostly responsible for tripping. And you can kind of like visualize like there's high concentrations right here, over there. To be very specific, you have the medial frontal cortex, the temporo-occipital cortex, the claustrum, stuff like that. And you'll notice that most of it is concentrated in the cortex, like on the outer shell of the brain. In case of emergency, yeah. If you're not having a good time. And philosophically, that's not my MO, because I think challenging experiences can be good experiences. I don't want to undermine that. Some challenging experiences, bad trips, can be traumatizing, and it might be useful to end a trip, but it just really depends on where you fall philosophically on the matter. Some people might say if you're having a bad trip, a challenging experience, you got to do the shadow work, you got to work through it, it's what you need, blah, blah, blah. Other people say that, oh no, there are certain dispositions that might make it so that a bad trip or a challenging experience is not something you want to be having at that moment. It's philosophical ultimately. All right, so pyramidal neurons. So these, yes. If you take it before a trip, you won't trip. And if you take it in the middle of a trip, it'll help bring it down. I don't know. I've never done it, so I can't answer that. Probably depends on how quickly your body can metabolize it and enter your bloodstream. Okay, yeah, pyramidal neurons. So layer 5 of the neocortex, there's a special variety of neurons that's responsible for tripping. That's these ones right here. This is what they look like. And the pyramidal neurons, they are basically where all of the output goes from the down level systems in your brain. Pyramidal neurons are basically where all of your bottom level signaling gets integrated in the brain. They are at the top of the hierarchy, and this is where a lot of your high level computational processing happens in the brain. They're very, very well connected. They're innervated. They connect to most regions of your brain. They're like the watershed where basically all of your signals go. And that's what makes tripping kind of like so holistic, so all-encompassing of your entire nervous system, is because your pyramidal neurons are like really downstream, and they are some of the most fundamental processing and computational units in your brain. And they typically fire at the alpha frequency. So neurons aren't if they're not they're not just chilling out at all times, not firing, they tend to have a baseline oscillation. The baseline oscillation of pyramidal neurons is the alpha frequency. Okay, this is a diagram. I really like this, but if you're not into neuroscience and stuff like this, it can be a really confusing and overwhelming diagram. But I'll try to explain it to you. So let me bring out the laser pointer. Okay, so this is a serotonin neuron, 5-HT, 5-hydroxytryptamine, that's serotonin. So this comes from your brainstem, and this serotonin neuron is projecting to, layers of your cortex that are deep, like 1, 2, 3, 4, 5. 1, 2, 3, 4. And then you have layer cortex 5, which is your deepest layer. This is where your pyramidal neurons are. So your deep cortical layers have some 2A receptors, and then serotonin is projecting to them. And then these deep cortical layers are also projecting to your layer 5 pyramidal neurons. So these ones have serotonin receptors, and these ones also have serotonin receptors. So these neurons have a finely coordinated firing pattern. They have a finely coordinated rhythm in how they fire. So your brain is really good at orchestrating ensembles of neurons, how they fire intrinsically. But what you do is when you introduce a psychedelic, it kind of just throws off the whole equilibrium. So you have your psychedelics that are stimulating these neurons, which are causing glutamate to be released, which are stimulating this neuron. So glutamate, remember, is the primary excitatory activator. So this neuron is all of a sudden getting a lot of glutamate because these neurons are being stimulated. And then this neuron is also getting serotonin and glutamate. So what this is basically doing is it's kind of like throwing off the finely tuned orchestration of the neurons, and it's kind of like causing anarchic patterns of behavior, patterns of firing. Anarchic, this term is going to come up a lot, anarchic firing. And the cool thing is, when these neurons are being stimulated with psychedelics, it causes an increase of what is called brain-derived neurotrophic factor, BDNF. And brain-derived neurotrophic factor actually causes neurons to grow. And this might be a mechanism of how psychedelics promote neuroplasticity. So when a neuron gets stimulated by a psychedelic, it causes some downstream effects, some downstream signaling. And one of those downstream effects is that the neuron will start producing more BDNF. BDNF is brain-derived neurotrophic factor. Neurotrophic is basically like nerves growing. So this diagram, it's kind of hard to see, but look at it closely. So what this researcher did, this is research that was done by the Olson lab. If anybody wants all the papers, contact me afterwards, message me, email me. I can send you all of the papers where all the research of this entire presentation came from. I'll send you the whole bibliography if you want it. But this research came from the Olson lab. So what they did was they did in vivo studies and in vitro studies. So basically what they did was they took some neurons, extracted them from an animal, kept them alive, and they put them in a petri dish. And what they did with those neurons in a petri dish is they stimulated those neurons in a petri dish with psychedelics. And the psychedelics they used were LSD, DMT, and another serotonin 2A receptor agonist called DOI. This is not psychedelic, but it stimulates the same receptor just so they could just tease out if the mechanism is involved with that receptor specifically. But anyway, so they had neurons in a petri dish, and they stimulated those neurons with these psychedelics, with these substances, and they found that, so imagine this is a neuron. The neuron had increased dendrite growth. So imagine that neurons look like trees, right? So the trees grew new branches. And then if you zoom in to one of the branches, so each one of these little notches right here, they're called neuronal spines, but this is where your synapses are. This is where neurons connect with other neurons. So you have increased dendrite growth, and you would increase spines, increase synapses. So the neurons grew new branches, and they grew new synapses. So this might be a mechanism that explains the neuroplasticity. So it basically allows neurons to grow new branches, and it allows neurons to form new synapses, new connections with other neurons. And this has a lot of relevance to depression, because what we see in people with depression is people who have long-term depression, it might cause atrophy of certain regions of the brain, meaning some synapses, some dendrites will actually atrophy, they'll wither away. So people with long-term depression, regions of the brain will literally wither away. And they've shown that with treatment with psychedelics, those regions of the brain that have withered away can actually regrow. Yeah, I basically just said that. So in the prefrontal cortex, which is one of the higher level regions of your brain that is really good for regulating your emotions, regulating your motivational states, regulating so much of what you can control cognitively. They found in fMRI studies that people with depression will actually have atrophy of the regions of this region of the brain. And the atrophy basically means, so when people typically hear the word atrophy in a medical setting, you think of muscle atrophy. So if you've ever broken your arm and you've been in a cast for like many months, after you get that cast removed, you'll find that your muscles are really weak. That's because when you don't use your muscles, if you don't use it, you lose it. Those muscles have atrophied. Similarly, the same thing happens in the brain. People with long-term depression, though some regions of the brain that are involved with emotional regulation, higher level cognitive control, those regions will actually atrophy. And it's been shown that with treatment with psychedelics, regions have atrophied, you can actually repair them. And the way you repair them is through this. So you get some regrowth of the branches and the synapses. Okay. So I've already kind of touched base on the fMRI studies. So basically what they do is an MRI machine is a magnetic resonance imaging. So the way it works, an MRI machine has a very powerful magnetic field. Everybody knows, well, maybe you don't, but blood contains hemoglobin. The way hemoglobin has its red color is because it contains iron. So the presence of blood is a good indicator of how much iron is present, and then we all know that iron is magnetic. So you can use an MRI machine to basically discern where there's extra blood flow, because it can detect the magnetic signal of all the iron in the hemoglobin. And when regions of the brain are more activated, they need more blood flow, they need more oxygen to sustain that activity. So you can kind of, assign a correlation or a proxy to brain activity by measuring the, it's called the BOLD response, the blood oxygen, what was that? Blood oxygen level dependent. Yeah, the blood oxygen level dependent response, exactly, thank you. So that's why MRI scanners are pretty useful. Of course, there are criticisms of fMRI. It doesn't have great temporal resolution, but you can kind of, get some data that shows you which regions of the brain are getting more blood flow, aka more activity. So they gave people LSD and they put them in an MRI machine and they scanned their brains. So they found that when people are experiencing visual hallucinations, they have a greater cerebral flow in the visual cortex. And RSFC stands for resting state functional connectivity. So they basically can analyze which regions of the brain are being activated concurrently and kind of create a network with each other. And this is called functional connectivity. And they find that when people are experiencing visual hallucination, there is increased resting state functional connectivity between the visual cortex and a lot more regions of the brain. So when people are having visual hallucinations on LSD, the visual cortex is communicating with many more regions of the brain than it typically communicates with. And this suggests that typically when your brain is in a sober state, your visual cortex is doing most of the heavy lifting in terms of creating the visual experience. But when people are having visual hallucinations, it suggests that more of the brain is contributing to the visual experience. And this also correlated with a decrease in the alpha signal. That's going to be important later. Alpha is a frequency of oscillation in the brain. So they've hypothesized that alpha frequencies are a way for the brain to kind of filter out signals that aren't super relevant. And I had mentioned that the innate firing pattern of the pyramidal neurons is at alpha. So when you flood the brain with all of these signals and you create anarchic firing patterns, you kind of like disrupt the innate alpha firing pattern. You disrupt the brain's ability to kind of like filter out a lot of the background noise. And it causes disinhibition, and it causes your neurons to kind of like have these anarchic patterns of firing where they're kind of like let off the leash. They're no longer filtering signals and regimenting signals in a way that's normal. And then when you get anarchic patterns of firing, you get visual hallucinations. So way back in the day, this researcher named Jack Cowan, he proposed that a lot of the visual hallucinations that people have while tripping tend to fall into certain patterns, certain motifs. Of them are lattices and honeycombs, cobwebs, funnels and tunnels, and spirals. And this can get into some really complicated physics, but basically the premise is, I'll give you this analogy. Imagine, this might be sensitive to say this in Boulder, but imagine a grass fire. So if you have a grass fire where all the grass is the same, it spreads out evenly, and it's just like meh. There's no pattern. It's just like an even spread of fire. But now imagine this grassy field has a bunch of grasshoppers in the field, and these grasshoppers, when they get hot, they start to sweat and they release a lot of water, and then this releasing of the water kind of like affects how well the fires can spread. If you apply this analogy to the brain, so you get anarchic signaling, anarchic firing of neurons in the brain, and then this firing causes some neurons that are inhibitory to send inhibitory signals, and then basically what you get is you get funky patterns with this. It's called a stochastic process, a stochastic Turing mechanism. I'm probably not doing the best job of describing this because this is not quite my domain, but in a way it's kind of like a mechanistic process for how these patterns can form organically just by anarchic patterns of behavior, patterns of firing. And one thing that's really cool is that a lot of this is entailed just by the physical anatomical structure of the neurons in your brain. So in many ways, the patterns you see, the visual patterns you see, I'll go back to the previous slide. If you look down in the bottom corner right here, a lot of these patterns you see are actually just you seeing the anatomical structure of your neurons, because the anatomical structure of your neurons, how they wire into each other, is kind of facilitating why these patterns emerge out of the anarchic firing of the neurons. Okay. Now we're going to get to the network level effects. So we just spent a lot of time talking about the micro level effects, but if we get to the macro level effects, the network level. So when serotonin activates, when the psychedelic activates your serotonin receptors, it disrupts the finely coordinated rhythm that your brain has kind of like orchestrated over the years. And by disrupting that finely synchronized and orchestrated rhythm, you disrupt the pattern and it desynchronizes the activity that your brain has kind of like cultivated over the years. And this orchestrator, so the central orchestrator of your brain, when it's no longer orchestrating, your basically lower level regions of the brain are kind of like let off the leash, and they're able to kind of like start firing in a way that is no longer orchestrated. And there are networks in the brain that have a finely tuned synchronization, and this finely tuned synchronization is no longer there. So there are regions of the brain that form networks, and these networks are basically no longer communicating with that in that synchronized manner. So that means the networks are disintegrated. Network disintegration. That's something that's crucial. So the networks are disintegrated. And one of the ways that researchers can measure this quality is by measuring entropy. So entropy, if you've heard that word before, it's a term in physics that kind of represents the opposite of order, the opposite of regimented, ordered sequences. Entropy is a measure of uncertainty, a measure of disorder. And then so when you no longer have the finely coordinated, synchronized orchestration, you have an increase of entropy in the brain. So in your brain, there are certain networks. So a brain network, one way to look at it is regions of the brain that are connected to each other, and they operate in concert, they operate in tandem. One notable brain network is called the task-positive network. So the task-positive network is active when you are doing a task that requires a lot of cognitive control. For example, if you are writing out math equations on a sheet of paper, you're using your task-positive network. If you're solving a Rubik's Cube, you're using your task-positive network. If you're doing something that requires you to focus and attend to it, you're using your task-positive network. And regions of your brain that might be part of your task-positive network are for example, your executive system, your motor system, your salience network. All these different networks are kind of like working in tandem to do the task. There's a different network in your brain that is inversely correlated with the task-positive network. What that means is that when the task-positive network is on, this network is off. Conversely, this network that's inversely correlated with the task-positive, it's called a default mode network. So conversely, when the default mode network is on, the task-positive network is off. So the reason why it's called the default mode network is because it's what's active at your brain at a default level. So when you're just hanging out, laying around, loafing, not doing anything in particular, the default mode network is the region of your brain that is going to be most active. And then the way to deactivate the default mode network is to do something. So when you get out and you start doing math equations, your default mode network is no longer running because they're inversely correlated. So the default mode network is generally active when you're idle and when your mind is wandering. Yes, those are just examples of brain networks.
Okay, but salience and those two don't correlate to others. Those are four different networks.
Yeah, they're different networks. Yeah, there's many networks of the brain that have, and they're all interconnected in many ways. But the crucial ones to note are the default mode network and the task-positive network. And the task-positive network incorporates both the salience network and the executive control network. So the default mode network is a very, very important, this is crucial. And a lot of the research coming out lately from the lab of Dr. Robin Carhart-Harris and a lot of the pop science books, like from Michael Pollan, give a lot of attention to the default mode network, deservedly so. And there is some controversy in the field regarding just how much clout we give the default mode network. I think it deserves the clout, but I don't want to get too much into the weeds of the drama in the scientific community right now. But basically, the default mode network is a very important network regarding some of the crucial effects of psychedelics on a network level. So because the default mode network is active when you're just like hanging out, not doing anything, being idle, it's a very central network of the brain. And it has a very high basal metabolic rate. So this network is drawing a lot of energy, even when you're just not doing anything. Because when you're just hanging out, loafing about, your brain is not off. Your brain is literally not off. You're being idle. Your mind is still wandering. Your mind is still going down paths. Your mind is doing things. So the default mode network is this network that's kind of responsible for all the patterns of mental behavior that occurs when you're sitting idle. And a lot of what it does. So just contemplate for a second. What does your mind do when you're just being idle? When you're just loafing around, your mind is wandering. What happens? Yeah, mental time travel. You start ruminating about the past. You start worrying about the future. You start remembering that embarrassing thing you said 5 years ago in your civics class. You start ruminating about all these things that are just kind of like on the back of your head, and you're kind of like keeping an inventory, keeping a catalog of where you stand socially among your peers. This is a sort of, it's a recurrent process that basically every human goes through, where you have these patterns of behavior, these patterns of thought, and a lot of them involve social relationships, and Robin Carhart-Harris and other researchers, they posit that the default mode network is the seat of the ego in the mind, in the brain. Because when you are mentally time traveling, you're thinking about that embarrassing thing you said, you're thinking about, you're worried about the future, you're thinking about your relationships. These are all things that are kind of like self-reflective. These are things that relate to your ego. So your ego is dwelling upon itself when you're loafing around not doing anything. And the default mode network is the network of the brain that is responsible for that. And I just want to reiterate and emphasize that the default mode network is a very central network of the brain. It's at the top of the hierarchy. The default mode network can control so many of the other networks of your brain, and it's being fed by so many of the different networks of your brain. If you look at like the network diagram of how different networks of your brain connect to each other, the default mode network is very central. It's a very high level network it's at the top of the hierarchy. So it's a central orchestrator of global brain function. It's connected to so many other regions of your brain because your ego is very very all-encompassing. Your ego controls your motivations. Your ego controls what you do with your life, what job you want to do, how you relate to others. Your ego is very crucial in dictating your behavior, and it's involved in metacognition. And there are certain regions of the brain that are kind of like the hubs of where your default mode network is located. The major nodes are your posterior cingulate cortex. So your cingulate cortex is, if you imagine the kind of like that midline of your brain. So like, your brain has two hemispheres, right? So I'll just draw this out. So your brain has two hemispheres, right? So your cingulate is like here, where your knuckles are. So your posterior cingulate cortex is the posterior, the back right here. And if you look at this little diagram right there, that's your posterior cingulate cortex. Your medial prefrontal cortex right here. So your prefrontal cortex is at the front of your brain. It's where a lot of your metacognitive and cognitive control and stuff like that. So these are nodes that are kind of like the seat of where your default mode network lives. And then you also have some medial temporal lobe. This is where your memories are. So you have these regions of your brain. So your central orchestrator that is connected with your memories and form a network. And these regions, the integrity of these regions is really heavily correlated with the integrity of your ego. And if you disrupt these regions, you have disruptions of the ego. And I will just highlight this really damn cool image that has been generated so this is a DTI, a diffuser tensor image. It shows how your different regions of your brain are connected by bundles of fibers. I love this shit. I love it. And like right here, like that's your medial prefrontal cortex, posterior cingulate, and your temporal lobes. Okay, so your ego. From the moment you're born, your brain starts to categorize the world and regiment it and create order out of the chaos. And in order to create order out of the chaos, your brain needs to put things in boxes. It needs to categorize things. And one of the ways it categorizes the world is it creates a model of itself. This model of itself is the ego. The default mode network has been posited to be the seat of the ego. Has anybody heard of the term a strange loop? Nice, we got a couple. Douglas Hofstadter wrote a whole book about this, a couple of them actually. But the idea of neurons kind of like turning inward and creating a model of themselves is something kind of trippy. It's really trippy. It's like taking a camera and then aiming the camera into the monitor and getting that recursive feedback loop. And that's kind of what happens when you have an ego. And it's just this really like mind-bending phenomenon. And you know, these images that I've incorporated into here, like a Mobius strip and that little animation, and this right here is what I just described. If you take a camera and you aim the camera into the monitor, you get these really trippy effects. And this is kind of like what your mind is doing when you have an ego. It's really trippy. So it gets even trippier. So for you to have an ego, for you to model your own mind, your mind is taking lots of reference frames. Your mind is incorporating models of how you believe other people perceive you. So let's look at Alice real quick. So here's Alice. Alice has a model of Bob, right? So Alice has a model of Bob. So she has a model of Bob, and in her model of Bob, she has a model of Bob's model of Alice. Similarly, Bob has a model of Alice, and Bob has a model of Alice's model of Bob. And this is true for all of your social relationships. For every single person you know, you have a model of them, and in your model of them, you have a model of their model of you. Every single social relationship, this is happening with your parents, with your schoolmates, with your coworkers, with that random person you walk by on the street who saw you trip. That person thinks I'm an idiot. Every single social relationship, you are keeping tabs of how that person sees you, what kind of esteem you have in their mind. And your ego kind of like functions as an aggregate of all of these different conceptions you have. Here's another way of illustrating it. There's Bob, his self-model, Bob's model of Alice, Bob's model of Alice's model of Bob. And then the default mode network is very heavily interconnected with the mirror neuron system. The mirror neuron system is activated when you see other people doing certain tasks. For example, if you see somebody throwing a ball, the neurons inside your brain responsible for throwing a ball are also activated. So there's this mechanism in your brain that kind of like reflects other people. And your default mode network is definitely very heavily intercorrelated with that. Here's another really cool thing I found on Reddit. I'll read it to you. A different version of you exists in the minds of everyone who knows you. I read a book that blew my mind. The main character goes crazy when he realizes no one really knows him. The gist is that the person you think of as yourself exists only for you, and even you don't really know who that is. Every person you meet, have a relationship with, or make eye contact on the street with, creates a version of you in their heads. You're not the same person to your mom, your dad, your siblings, than you are to your coworkers, your neighbors, or your friends. There are 1,000 different versions of yourself out there in people's minds. A you exists in each version, and yet your you, yourself, isn't really someone at all. So as you can see, this ego nonsense can get really fucked up. I love this quote. We are not who we think we are. We are not who others think we are. We are who we think others think we are. Imagine if you lived out in the woods, didn't know a single person, and you lived like that your whole life. Would you have a conception of yourself? Would you have an ego? Ponder upon that. So as you can see, the default mode network, the ego, an overactive default mode network can be responsible for some pretty nasty things. If you're ruminating on all the embarrassing things that have happened to you all the time, if you're over-obsessing about every little embarrassing thing you've done, if you're thinking about every single person who dislikes you, every single person has a negative perspective on you, every single little bit of humiliation and embarrassment you felt, all these things feed into each other. The ego ruminates on these things. And if it's constantly ruminating upon these things over and over and over again, this creates some pretty nasty feedback loops in the brain. This can create jealousy and envy, creates pride and humiliation, results in depression and anxiety, and even it's fear of death. Because you've grown this ego, you've gone attached to what happens when this self is gone. The ego is all you've ever known. Ever since you were born, your brain has created this model of the self. And then what happens when this model is gone? Cessation, death. It creates fear of death. Here's some really cool art pieces by Alex Gray that kind of illustrate some of the negative affective states that come from an ego that is out of equilibrium. You have self-hatred. I think these are really potent, powerful illustrations. You have despair. Here's another meme of what happens when you have an overactive ego. Not caring what people think about you. That's, you don't care. You sometimes think about what people say to you. Or you think a lot about when someone insults you. Or you stay up until 3 a.m. in the morning thinking about every little mistake you have ever made and how you could have done that situation better. Who can relate to this? This is the ego. This is the default mode network. Okay, so psychedelics cause disintegration of the default mode network. So I had mentioned earlier that your cortex, your neocortex, the networks of your brain, these regions of your brain that are interconnected, they have a finely orchestrated pattern of behavior. They have a finely orchestrated system of synchrony and coordination. And then when you take psychedelics and then you disrupt this finely coordinated orchestration, these networks that are tightly coordinated, tightly synchronized, these networks disintegrate. And they're no longer able to coordinate and create this cohesive model of the ego. And this sets off a cascade, because I had mentioned that the default mode network is a very highly central network. It's at the top of the hierarchy. And if you disrupt this network's ability to orchestrate the rest of the downstream networks, you kind of like let the networks off the leash. The default mode network can no longer hold cohesion, and you get network disintegration. I had mentioned that. And you also get network desegregation. So what network desegregation means is that networks typically know who they're communicating with. They have this set pattern of behavior of this network is going to hook up to this network, send signals to that network, and they have orchestrated pattern of behavior. But when you have network desegregation, networks that typically don't communicate with each other all of a sudden start communicating with each other. So psychedelics, they reduce the stability and integrity of well-established brain networks. They induce network disintegration and desegregation, and the cortical brain activity becomes more entropic. These are some of the fundamental principles to take away about how psychedelics affect brains on a network-wide level. So I'd shown you that diagram earlier, that cool diagram that I liked of the DTI imaging. And you have the posterior cingulate cortex right here, and you have the medial temporal lobe. So the posterior cingulate cortex, it's a hub where information about your spatial world gets integrated. And then it's also a hub where information about your memories and emotions kind of flood in, and your medial temporal lobes are involved in your memory and emotion. So your medial temporal lobes are feeding into your posterior cingulate cortex, kind of like creating this model of your body and your ego. And when the psychedelics are introduced into the body, it disrupts the finely tuned orchestration between these two regions of the brain, and it causes them to decouple. And when you decouple them, they can function independently of each other. So usually these regions are sending signals back and forth between each other, orchestrating each other. When you have disrupted this coordination, this synchrony, they are now decoupled. They're able to operate independently of each other. And this can cause a collapse in your sense of separation between self and other, and between your perception of inner space and outer space. And some people call this the feeling of oceanic oneness. So disintegration of your default mode network correlates with ego dissolution. Everything has been leading up to this. So in the lab, when they put people in the fMRI machine, the scanner, and they kind of figured out which regions of the brain were correlated with, which reports. So while people were in the scanner, they had questionnaires. Every 15 minutes, they would fill out a questionnaire reporting how they were feeling. And then when people were reporting feelings of ego dissolution, or they don't feel like they have a sense of self, or they feel this feeling of oceanic oneness, they had found that this had correlated very strongly with disintegration of the default mode network and decoupling of the, I think that's the retrospinal cortex, and I forget what the pH is. Maybe that's in my notes, but I'll dig through the article if anybody's interested in that. But anyway, basically the premise is when your default mode network disintegrates, your ego disintegrates. And this creates the famous mystical experience of psychedelics. They call it the unitive experience or oceanic oneness. It's all-encompassing unity or oneness, and it's a collapse in some of the most fundamental dualities we carry with ourselves. This sensation of self versus other is gone. This sensation of subject versus object is gone. This sensation between internal versus external is gone, because these regions of your brain are so preoccupied with regimenting the world, kind of creating this distinction between this organism and the rest of the ecosystem, the rest of the environment. And this happens through the finely coordinated brain activity of all these different regions. And when you disrupt this mechanism of your brain regimenting itself away from the environment, away from the universe, you've disrupted this regimented categorization of your brain networks, and then you have released anarchy in the brain. And these systems, these models, these modules of the brain, they collapse. And you're left with the oceanic oneness feeling, the mystical experience. This can also be elicited by stroke. So I'll read this to you. On the morning of December 10th, 1996, Dr. Jill Bolte-Taylor suffered from a massive left hemisphere hemorrhage as a result of congenital blood vessel malformation. She says, "I remember that first day of the stroke with terrific bittersweetness. In the absence of the normal functioning of my left orientation association area, my perception of my physical boundaries was no longer limited to where my skin met air. I felt like a genie liberated from its bottle. The energy of my spirit seemed to flow like a great whale gliding through a sea of silent euphoria. This absence of physical boundary was one of glorious bliss." So when people have a stroke, regions of their brain are deprived of oxygen, they're no longer able to do their job, you get the same sort of disinhibition. So regions of the brain are no longer able to do their job of regimenting and saying, oh, this box, this organism is categorized separately from that, and you get this oceanic feeling. So when you get different networks let off the leash, the central orchestrator is no longer in control. Rest of the brain is let off the leash and able to communicate with other regions they don't normally communicate with, and they have crosstalk. And this image got really famous back in the day. So here's placebo. So people are given, so what this diagram is every one of these little dots is a region of the brain, and each one of these lines is a connection between one region of the brain to another region of the brain. So in typical baseline consciousness, people that got a placebo, this is how their brain connectivity motifs showed up. You know, you have this standard repertoire of brain connectivity. When you give people psilocybin, the repertoire of brain connectivity is increased. So regions of the brain that typically don't communicate with each other because their signaling is regimented and orchestrated, and they have these finely tuned patterns of activation and connection and communication. Those finely tuned patterns of connection and communication are disrupted. The brain is let off the leash, and regions of the brain are now able to freely mingle with other regions of the brain. And this is measured as an increase in entropy, because the typical ordering of your brain prior to this is, because it's highly regimented, it's predictable, your brain has a comfortable pattern of behavior, a comfortable sequence of motifs of activation. This orchestrated system, it knows it well. It's highly predictable. And then when you introduce uncertainty, when you introduce all of these crazy interconnectednesses, it creates entropy. So entropy is a measure of uncertainty. So Dr. Robin Carhart Harris, he has proposed a fundamental model of the quality of consciousness that he calls the entropic brain hypothesis. And basically, the crux of it is that your brain is kind of like always at the razor's edge of order and chaos. So your sensory environment is always, it's like this chaotic soup of information and sensory input, and your brain wants to make sense of this chaotic soup, so it tries to do whatever it can to create order out of the chaos. And your brain is kind of like always at this razor's edge state between order and chaos. And he uses a terminology called primary consciousness versus secondary consciousness. So I'll skip primary for now. So secondary consciousness is what your brain goes through when it's sober. And it's called secondary consciousness because your brain has to learn how to be sober. Your brain has to learn how to have that hyper-regimented, controlled, ordered state. Primary consciousness is chaotic. It's entropic. It's infant-like. So when you're first born, before you've learned how to regiment the world, before you've learned how to develop an ego, you are in this primary state of consciousness, which is very chaotic, it's very entropic. Aldous Huxley calls it the mind at large. And as your brain develops, your brain learns how to minimize entropy. Your brain does not like entropy. Your brain does not like uncertainty. Your brain does not like disorder. Your brain wants order. Your brain wants the ability to predict things. Your brain wants to minimize entropy and form rigid, ordered structures and networks. And that leads to the transition from primary consciousness to secondary consciousness. Secondary consciousness is ordered, it's predictable, it's regimented, it's regularized, it has patterns. So let me know if you can't read this. So primary states of consciousness are marked by high entropy. Secondary states of consciousness are marked by low entropy. So low entropy, so entropy just means disorder. So low disorder, so your states of consciousness are very rigid. They're very solid, they're very ordered, they're very patterned. High entropy states have high disorder. They're very flexible, they're not rigid. So there are some examples of primary consciousness. One of them is the psychedelic state. Another one is infant consciousness. Another one is REM sleep and dreaming. You have psychosis, sensory deprivation, the near-death experience, magical thinking, the dreamy state of temporal lobe epilepsy, and finally, divergent thinking or creativity. So these states are all marked by being high entropy, high disorder, and the very flexible states of consciousness. To be an artist, to be creative, you have to disrupt patterns. You have to have novel states of thought that are out of the typical regimented patterns of people. And then on the other side of the coin, you have states of consciousness that are marked by low entropy, low disorder, hyper-rigidity, coma, anesthesia, sedation, deep sleep, seizure. And here's where it gets interesting. Depression. When you're depressed, your consciousness is hyper rigid. You're constantly ruminating on the same patterns of thought, the same patterns of rumination and self-loathing and stuff like that. OCD. People with OCD have hyper rigid compulsions, patterns of behavior. Addiction. Another hyper rigid pattern of behavior. These things are hard to unshake. Your brain likes the order of having these compulsive drives to consume substances. Rigid, narrow thinking. People who are stuck in their mindsets, stuck in their ruts of thought. People who are closed-minded. They're very hyper rigid. So your brain is kind of like right here. So your normal waking consciousness is like chilling right here. It's kind of like at the cusp of high entropy and low entropy. So if you're marked by something like depression or anxiety, you are kind of like edging your way in this direction into a state of hyper rigid thinking, low entropy, low disorder. When you have a psychedelic experience, you kind of regress from secondary consciousness back into primary consciousness, back into that infant state, back into that dream state, back into that divergent creative state, back into that entropic state, high entropy. So this brings us to mental ruts. People who have depression, for example, they're stuck in a rut. When people are depressed, they're constantly reliving the same humiliations, the same terrible things, the same depressive things, and their minds are stuck in these loops. Their minds are stuck in this deep rut of I can't escape. I can't escape these patterns of self-loathing, these patterns of depression, these patterns of ahedonia, and you're caught in that rigid state. People with anxiety too. If you're anxious, your brain is marked by this rigidity, this recurrent pattern of worrying about the future, this recurrent pattern of having a sympathetic nervous system response. These patterns are marked by, they're highly ordered. So it seems odd to describe these states as being highly ordered, but your brain falls into that rut of regularity, it falls into that rut of order, it falls into that rut of low entropy. And the way that psychedelics help is they induce this ability to escape from the rigid grooves of accumulated mental experience. So I'm going to show you a visual analogy of this later on, but when you have recurrent patterns of thought, recurrent patterns of behavior, this sort of like digs grooves in your mind. You can call it, the scientists call it deep attractor valleys. And the more you have a recurrent thought, the more you have recurrent behaviors, the more you have recurrent patterns, the deeper those grooves get, and those grooves become deep ruts because your brain loves the order. Your brain loves the predictability. Your brain loves the consistency. So I'll try to describe this. So in your brain, you have two parallel pathways of processing. You have your bottom-up processing, which is your raw sensory data, stuff that you're just soaking in from the environment. And then you have top-down information flows, which is, let's just say the top-down would be your default mode network, kind of like orchestrating and regimenting the rest of the system. When you have hyper-rigid thinking, when you have an overactive default mode network, when you have things to that effect, your top-down signals are very strong. And your top-down signals influence how much of your bottom-up signals can make it into your consciousness. And then this creates a deep groove, a deep landscape, is what I'll call it. So what psychedelics do is they decrease the weighting of the bottom-up signals. So the top-down signals can usually regiment how much of the bottom-up signals can enter your consciousness. But if you disrupt that, the bottom-up signals are kind of like let off the leash, and they're no longer inhibited and controlled by the top-down signals. This is another way of illustrating it. So imagine this ball, is novel sensory information from your environment. In your typical sober consciousness, it lands and it doesn't really leave a ripple because your top-down consciousness, your top-down networks and stuff like that can regiment how much of the bottom-up information flow can enter your consciousness. In the psychedelic state, the sensory input can leave ripples because your top-down modules no longer have the same influence to regiment the ability of your bottom-up to influence your brain and your consciousness. So one way of using a visual analogy to describe this is by imagining a ski slope. So a ski slope, imagine every time you have a thought, imagine every time you have a pattern of behavior, a pattern of rumination, a pattern of anxiety, a pattern of any of this, addiction, OCD, depression. Anytime you have a very regimented state of thought, state of being, every pattern of thought leaves grooves. And every time you have repeated patterns of thought, those grooves get deeper. Imagine you're at the top of a ski slope. Somebody pushes you down the ski slope. Where are your skis gonna go? Your skis are gonna follow the grooves that are already carved deep. This is how your brain works. So your brain loves the regularity of always following the paths that it knows and has that predictability, has that regularity. What psychedelics do is they can basically create a snowfall where those deep grooves are leveled out and your brain has the ability to carve new paths and get out of the rut and form new grooves. So here's another cool thing. Experienced meditators. So we've talked about how the dissolution of the default mode network is predictive of ego dissolution and people being able to escape deep mental ruts. Seasoned meditators, they've scanned the brains of seasoned meditators and they've shown that they have reduced activity in the default mode network. So there are ways that mindfulness meditation can kind of lead people to cultivate a baseline mental practice that it can sort of approach a similar principle of not letting the default mode network have as much of a grip in forcing people to fall into these same regular patterns of thought, these same regular patterns of behavior. And we're almost at the end of the presentation, and I will just say that while, yes, psychedelics can help people kind of like shake the snow globe and form new patterns of behavior, new patterns of thought, new patterns of ego. And there's a formative period after the psychedelic experience where the snow is fresh and you can carve new pathways in your brain. But if you fall back into the same patterns, if you fall back into the same environmental conditions, if you fall back into the same old, your brain will carve those same grooves. So there's a formative period immediately after a psychedelic experience where the snow is fresh and you have the ability to lay down the new grooves. But if you don't seize the opportunity to lay down those new grooves, and if you fall into the same patterns of behavior, the same habits, the same environments, your brain is just gonna lay down those same grooves as before. So that's why that formative period immediately after a psychedelic experience is so crucial into laying down those new grooves. Integration, yeah, psychedelic integration, that's the buzzword. Cool, well, that's the end of the presentation. Thank you. Thank you, everybody. And one more thing. So this is really exciting. We have some local decriminalization efforts going on. So right here in the city of Boulder, we are trying to get psychedelics decriminalized here in Boulder. And we are basically doing this by appealing to city council. So just to give you a little background about myself, I used to live in Ann Arbor. I went to University of Michigan, and when I was living in Ann Arbor, there was a group called Decriminalize Nature Ann Arbor, and we came together and we basically followed the model of Oakland and Santa Cruz, who were the first two cities to decriminalize a set of psychedelics. Denver was the first city to decriminalize mushrooms, which was huge. So shout out to Denver. They decriminalized mushrooms, was it in 2018 or 2019? 2019. So yeah, Denver was the very first city in the US to decriminalize psilocybin. And then shortly thereafter, Oakland, California decriminalized the natural psychedelics, followed by Santa Cruz. Number 3 was Ann Arbor, Michigan, and I was a part of that project in Ann Arbor, Michigan. And the way we did that, the way we got Ann Arbor to successfully decriminalize natural psychedelics was basically just appealing to city council. We would get members of the community to email city council. We got members to send them video testimonials, and we would show up at city council meetings and just bug the shit out of city council. We finally got them to vote on it, and when they voted on it, it passed. Any guesses? 11-0. It was unanimous. So we're trying to keep that same momentum going on here in Boulder, and in order to do that, I'm helping organize people who are interested in showing up at open comment at city council meetings. So I'm organizing a push for people to show up at the city council meeting this upcoming Tuesday, June 7th. So if you're interested, if you live in Boulder, and if you're interested in telling your story, why you believe psychedelics should be decriminalized here in Boulder, if you can prepare a 2-minute statement, sign up for open comment at City Council. If you're interested in doing that, come speak to me afterwards. I'm going to be hanging out. Come speak to me. We'll exchange information, and I'll coordinate with you, and we'll try and get you on the roster, on the lineup for open comment on the City Council meeting this Tuesday. It works. When you city council people, the city politics works entirely different than state politics. City council members, I'm pretty cynical about state and federal politicians. A lot of people share my cynicism, but when it comes to city politicians, they tend to be members of the community. They tend to be neighbors. They tend to be people that care about stories. The strategy is we open minds by appealing to the scientific research, we open hearts by appealing to the personal testimonials, and then we ground it by appealing to the long, ancient, indigenous tradition of these medicines on the land. So if you're interested in sharing your story, why you believe psychedelics should be decriminalized here in Boulder, come speak to me. We'll coordinate. We'll get you to be on the lineup for open comment on Tuesday, June 7th, and yeah. Secondly, we are, yes?
I just wanted to say I have the ballot for Initiative 61. So if you guys want to come and sign it, I'll support. I have it.
Cool. Yeah. So speaking of statewide ballot Initiative 61, so yeah, we're also working on the statewide level. So we have crafted a ballot initiative that removes 5 substances from the Controlled Substances Act. It's psilocybin, psilocin, DMT, mescaline, and ibogaine. And we're currently collecting signatures. And if we cross that threshold, if we get, we're shooting for, we need a little bit more than 130,000 signatures. And if we get that by August 8th, then we get on the ballot for November here in Colorado. And if you know how Colorado votes, we love our freedom, we love our liberty, we love our medicine. If it gets on the ballot, it's highly likely to pass. So we are currently collecting signatures. If you're interested in volunteering to collect signatures, we need as many people as we can to collect signatures. Come speak to me. And I personally advocate for Initiative 61. There's another initiative out there called 58, which creates a highly regulated medical model that gives the authority to the state of Colorado to regulate. I don't think we can trust the state to impose their regulations quite yet. I believe in a decriminalization first, a liberty model that allows people to have whatever relationship they want with the substances. If people want to take mushrooms in their backyard by themselves, they should have the right. If people want to have ceremony in a group, they should have that right. If people want to microdose, they should have that right. If people want to have a psilocybin experience in a medically supervised setting, they should have that right. But however, the last thing I mentioned, psilocybin in a supervised medical setting, if that comes first, then the lobbyists are going to come in and try and use their leverage to rig the system in a way that creates scarcity and tries to stomp out competition for their bottom line. So while I do believe that people should have the right to have psychedelic experiences in that medically supervised, controlled, and regulated setting, I believe that decriminalization, full, complete decriminalization, has to come first. And that's why I am a supporter of statewide ballot initiative 61. We have the ballot, we have the petitions here, so we'll be collecting signatures. And yeah, and thank you. And if you found any value in today's lecture, this presentation took a lot of work. If you found any value in the presentation, that's my Venmo. I appreciate any contribution. I'm sure most of the people in the room bought tickets. If you stumbled in this room without tickets, this was a paid event, so I would appreciate any contribution and anything if you have the ability. And thank you so much. And I'll be sticking around after the fact and talking and chatting, and we'll be collecting signatures for ballot initiative 61. And thank you. My name is Ramzy Abueita, and I hope you guys enjoyed the presentation.